Hi, my name is Azmain, and today I'll be talking about a bioconjugation strategy for delivering proteins using lipid nanoparticles. In the drug development space, protein-based therapeutics comprise a rapidly growing subset of pharmaceuticals. However, due to their large hydrophobic structures, delivery of proteins across cell membranes has been a long-standing issue in the field. And as such, protein-based therapeutics are currently limited primarily for extracellular applications. To overcome these delivery issues in the hopes of translating protein-based therapeutics for a wide range of intracellular applications, we were interested in adopting clinically proven delivery strategies established for other biological drugs. To this end, we turned our attention to lipid nanoparticles that have been developed for the delivery of nucleic acid therapies. Researchers have shown that if nucleic acids, which are negatively charged, are mixed with cationic lipids, they can become electrostatically entrapped within hydrophobic nanoparticle structures, which can then allow for nucleic acids to enter cells. To use these same nanoparticles for protein delivery, the hypothesis we wanted to test was pretty simple. What if you could mimic the anionic nature of nucleic acids by chemically modifying protein surface charge? Specifically, we were interested in designing lysine reactive linkers containing a cleavable disulfide bond, which can then be attached onto the surfaces of protein cargos. This will allow for electrostatic complexation with cationic lipid reagents, which can then lead to the delivery of protein cargos into cells. To test our theory, we constructed a panel of 3 sulfonate-containing linkers and used superfolder GFP as a model protein to study delivery with. Successful linker conjugation can be visualized by running the proteins through a gel across an electrical gradient. Here we can see that SFGFP reacted with increasing molar equivalencies of the 3 sulfonate linkers causes the proteins to run faster towards the positive potential end located at the bottom of the gel, indicating anionic modification. Incubating these same protein samples in the presence of glutathione, a compound that is found at high concentrations inside of cells, causes the protein bands to collapse back up towards the top, suggesting that the linkers can be cleaved off and can allow for proteins to revert back into their native form. After confirming linker conjugation, we next wanted to see if we could deliver anionically modified SFGFP using a commercial cationic lipid reagent called lipofectamine, which is mainly used to transfect DNA and RNA into cells. From flow cytometry data shown here, where fluorescence intensity is on the x-axis and cell count is on the y-axis, we see a shift toward higher fluorescence in cells incubated with lipofectamine and anionically modified SFGFP, indicating that we have successful delivery. In cells treated with lipofectamine and unmodified SFGFP, shown here in red, we observe a minimal shift in fluorescence, which tells us that delivery is solely due to anionic modification. Evidence of delivery can further be confirmed from confocal microscopy images, where we see clear internalization of protein only in cells treated with lipofectamine and linker-modified SFGFP. Next, we wanted to explore if proteins could be delivered using clinically validated lipid nanoparticle formulations, which are typically used for in vivo delivery of nucleic acid therapies. For these studies, we formulated lipid nanoparticles using varying amounts of a lipid called MC3, as shown here. For nucleic acids, lipid nanoparticles are usually formulated in acidic buffer, but for proteins, we wanted to see if nanoparticles could form around the proteins under more tolerable pH ranges. Additionally, we wanted to see if using a modified formulation consisting of an extra cationic lipid called DOTAP would aid in nanoparticle formation. From flow cytometry data using anionically modified SFGFP, we see robust protein delivery with the nanoparticles that are formed with DOTAP, and the observed fluorescence shift is even greater than that found with proteins delivered with lipofectamine, as shown here in green. We also repeated these experiments for unmodified SFGFP and saw no observable signs of delivery. Finally, we find that proteins delivered with lipid nanoparticles exhibit negligible cytotoxicity, with most cells showing viabilities above 90%. To summarize, in this work, we've shown that anionic bioconjugation of proteins can enable intracellular protein delivery with nanoparticles. With that, I'd like to thank you all for stopping by to hear me talk about my research.